Welcome. In this presentation, we're going to look at the structure of nucleic acids, DNA and RNA. We'll look at how they're assembled and take a brief preview of how they do their job of coding and transmitting information for the synthesis of proteins. You're probably familiar with images of DNA that look something like these pictures here. You're also likely familiar with the fact that DNA consists of a molecular code. So let's look at how the structure of DNA at the molecular level allows for the encoding of information. Let's start with this diagram here. Each of the different colors that serve as rungs in this ladder is an individual subunit of DNA. Also, notice that each rung of the ladder has two parts, or two colors, here in this diagram. As we look at the details of how DNA is constructed, we'll be looking at only one side of this double helix. In the diagram on the left, we're looking at a single strand of DNA. This single strand of DNA is formed by the polymerization of single nucleotide monomers. Each monomer is, in the middle panel here, consists of a phosphate group, a sugar, and a nitrogenous base. The sugar and the phosphate group are the same for every single nucleotide monomer, in the same way that the amino acid backbone was the same for every amino acid. Let's look a little bit more in detail at the components of a nucleotide. Focus on the red sugar in the middle. This is the 5-carbon sugar, and the carbons are numbered starting on the right. The third carbon has a hydroxyl group attached to it, and the 5th carbon is off the ring structure and has the phosphate group attached to it. Just like we've learned before, polymerization happens through a dehydration or condensation reaction. The results of this reaction is a dinucleotide and a water molecule being expelled. In DNA polymerization, always happens at the 3' end. That means that the chain lengthens at the 3rd carbon on the sugar subunits. Nucleotides are never added to the phosphate group at the other end of that chain. As the nucleotides are assembled at the 3' end of a growing polynucleotide, you can notice that the backbone consists only of the phosphate group and the sugar group, and for this reason we call it a sugar-phosphate backbone. The nitrogenous bases here, shown in green and blue, play no role whatsoever in the backbone. Lastly, notice that this strand of DNA has directionality. There's a 5' end that's located at the phosphate group, which is attached to the 5th carbon of the sugar. There's also the 3' end, which is the unattached 3rd carbon of the sugar group. As I mentioned, DNA always polymerizes at the 3' end. Now, let's take a look at the nitrogenous bases. In DNA, we have 4 bases: cytosine, thymine, guanine, and adenine. 2 of these, cytosine and thymine, are pyrimidines with only 1 ring. Guanine and adenine are purines, and they have 2 rings. One way to help you remember this is that guanine and adenine both have the word 9 in them. The purines each have 9 membered rings. Notice that there's a 5th base here. This is uracil, which is a pyrimidine, but it only exists in RNA, and it essentially takes the place of thymine, which exists in DNA, but not RNA. Remember when we looked at proteins, we found that there were different levels of structure. The primary structure of proteins was simply the sequence of amino acids. Similarly with DNA, the primary structure of DNA is the sequence of nucleotides. The double helix of DNA is formed from the secondary structure. Here, we can see one strand of DNA going, as the arrow points, in the 5' to 3' direction, going down the page on the left. Next to that is a similar strand of DNA also going in the 5' to 3' direction up the page. The second strand is going in the opposite direction to the first. We term this anti-parallel because the strands are parallel to each other, but they run in opposite directions. Between the backbones, we have the nitrogenous bases. The nitrogenous bases of one strand interact through hydrogen bonds with the nitrogenous bases of the other strand. The pairing of these nitrogenous bases happens in a very specific manner. The distance between the backbone strands is determined by the base pairing within that double helix. Each pair of bases consists of a purine and a pyrimidine. If two purines were to try to bond together within that space, they would simply be too large, and they would force the backbone strands apart. Two pyrimidines would take up too little space, and they would force the backbone closer together. Empirically, we find that the distance between the two strands is consistent and constant throughout the length of the DNA, indicating that a purine is always paired with a pyrimidine. Guanine and cytosine have 3 atoms that are able to form hydrogen bonds across the DNA helix. Adenine and thymine only have 2 atoms that are able to form hydrogen bonds. As a result, guanine always pairs with cytosine, and adenine always pairs with thymine. Even though we draw DNA as a ladder, as shown on the left, we need to recognize that it does twist into a double helix. The strands don't twist evenly like a rope. Instead, they form a major groove with more space and a minor groove with less space. Figuring out this structure in 1953 earned Watson and Crick the Nobel Prize. In many respects, RNA is similar to DNA. The differences come about due to the lack of a hydroxyl group on the second carbon in the DNA, but present in RNA. That DNA is deoxyribonucleic acid, whereas RNA has the oxygen and is therefore ribonucleic acid. Another difference between RNA and DNA is that, as I already mentioned, DNA has thymine, whereas RNA has uracil. Like thymine, uracil can form hydrogen bonds with adenine. One of the consequences of these differences in structure is that RNA does not form a double helix in the same way that DNA does. However, the nitrogenous bases in RNA can, and do, form hydrogen bonds in the same way as DNA. Therefore, part of the RNA strand is single-stranded, and parts loop back on themselves to form double-stranded regions. A consequence of not having full base pairing, as with DNA, is that RNA is much less stable and much more reactive than DNA. So, what role do DNA and RNA play in the cell? DNA is the molecule onto which all genetic information is stored. It needs to be kept safe and as unchanged as possible. In eukaryotic cells with a nucleus, DNA is stored safely inside the nucleus and is never able to come out into the cytoplasm, where there are a number of chemicals that could damage it. So, DNA is like a precious book in the library. It doesn't do anything by itself, but it contains a lot of information for how to operate and build a cell. It gets this information out into the cytoplasm by way of messenger RNA. Messenger RNA has the same code as portions of DNA called genes. In fact, the DNA writes the code directly onto the mRNA. mRNA, unlike DNA, is able to exit the nucleus. The information on the mRNA is then used by ribosomes to make the primary structure of proteins. We'll learn later in the course how the genetic code is translated into a protein code. This idea that information flows from DNA to RNA to protein is known as the central dogma of molecular biology. Notice that the information flow is only in one direction, from DNA to RNA and finally to protein. It used to be thought that there was no mechanism for information going in the other direction. However, more recently, we have learned that information can be written from mRNA back into DNA. In fact, this is what retroviruses do. They cause information on their RNA genome to be written into the DNA of a host cell.